Hey everyone, in this lesson we are going to learn about Tricarboxylic acid cycle or TCA cycle, or it is also known as Citric Acid Cycle, or it is also known as Krebs cycle. Before I talk about citric acid cycle, I first want to give you an overview of cellular respiration. And cellular respiration occurs in three major stages. First stage is the production of acetyl-coenzyme A, and it begins in cytosol, but acetyl-coenzyme A is eventually formed in the mitochondria. So acetyl-coenzyme A can be synthesized from different organic molecules such as amino acids, carbohydrates, and lipids. And all these fuel molecules, when they oxidized, they have one common end product, that is acetyl-coenzyme A. Now, I haven't covered amino acids and fatty acid oxidation, but I have covered how glucose is converted into pyruvate through glycolysis, and just in my last video I have talked about the mechanism of oxidative decarboxylation of pyruvate to acetyl-coenzyme A. So this is the first stage, which is the production of acetyl-coenzyme A. Now second stage is the oxidation of acetyl-coenzyme A, which completely takes place in mitochondria. And in this stage, the acetyl group from acetyl-coenzyme A is oxidized into carbon dioxide, and the electrons which are liberated through these oxidative processes, they are then passed on to the coenzymes such as NAD plus and FAD, and these coenzymes are then reduced to NADH and FADH2. So this is the second stage, which is the oxidation of acetyl-coenzyme A, and this is what we are going to learn in today's lesson, which is the citric acid cycle or Krebs cycle. And in this stage, electrons which are carried by these reduced coenzymes, they are then transferred to the series of electron carrying molecules of electron transport change, resulting in the formation of water. And the electrons, when they are being transferred, they actually create this proton gradient across the membrane, and this proton gradient is then used to drive the synthesis of ATP. So this is the third stage, which is the respiration, and I will have a separate video on electron transport chain in my next lesson. So today we are going to talk about citric acid cycle or Krebs cycle. So I just want to give you a brief basic information about citric acid cycle. First thing, it is the final pathway where the oxidative metabolism of carbohydrate, amino acid, and fatty acids converge into the synthesis of acetyl-coenzyme A. The oxidation of acetyl-coenzyme A provides energy for the production of majority of ATP. So this energy are provided in the form of these reduced coenzymes, which are then transferred into the electron carrying molecules of ETC to synthesize ATP. Now third, citric acid cycle totally occurs in the mitochondria, which is really important to know. And fourth, TCA cycle is an aerobic pathway because it requires oxygen. Now, TCA cycle was discovered by the German-born biochemist whose name was Sir Hans Krebs, and to honor his work, TCA cycle is also referred to as Krebs cycle. So there are 8 reactions of citric acid cycle, and we are going to learn about this each and every reaction in detail. So first reaction is the synthesis of citrate, which is also the condensation reaction, and this is the first irreversible reaction of the citric acid cycle. In the first reaction, acetyl-coenzyme A condenses with oxaloacetate to synthesize citrate. Here you can see that acetyl-coenzyme A has 2 carbon atoms and oxaloacetate has 4 carbon atoms, and then when these two condenses, they form citrate, which now carries 6 carbon atoms. And the reaction is catalyzed by the enzyme known as citrate synthase, and the reaction also releases coenzyme A. Now the product of this reaction, which is citrate with 6 carbon atoms, is also a type of tricarboxylic acid because it carries these 3 carboxyl functional groups. So that's why it is also called as tricarboxylic acid. Now the first reaction of citric acid cycle is irreversible reaction, and therefore the citrate synthase enzyme is also under the tight regulation. So let's talk about the activators and inhibitors of citrate synthase enzyme, and let's first talk about the inhibitors. For instance, if there is too much of acetyl-coenzyme A being synthesized, it is then going to be oxidized, and the electrons which are carried through these reduced coenzymes, they are then going to be passed through electron transport change to synthesize ATP. So basically end product is the synthesis of ATP. So higher cellular content of ATP can directly inhibit the citrate synthase enzyme. And I have always mentioned that increase in cellular ATP content is an indicator of cells with energy abundance. And second is the increase in the cellular level of NADH. And we know that if there is too much of acetyl-coenzyme being oxidized, it is going to generate more NADH. And this NADH can also inhibit citrate synthase enzyme. Now next is the product of this reaction, that is citrate itself can inhibit citrate synthase enzyme. So if too much of acetyl-coenzyme is being oxidized, that means it is also going to synthesize too much of citrate, and increase in the citrate level in the mitochondria can itself inhibit citrate synthase enzyme. Now last is the succinyl coenzyme, and succinyl coenzyme is the downstream product of citric acid cycle, and that itself can inhibit citrate synthase enzyme. So these are all the inhibitors of citrate synthase enzyme. Now let's talk about the activators. So increase in ADP levels in the cell can activate citrate synthase enzyme. Increase in the cellular ADP levels is an indicator of cells with energy deficient. That means cells require more energy, and therefore increase in the ADP levels in the cell activates citrate synthase enzyme. So acetyl coenzyme is oxidized fully to synthesize ATP. Now second activator is acetyl coenzyme. So if cells need energy, acetyl coenzyme can activate citrate synthase enzyme to synthesize more ATP. So this regulation of citrate synthase all depends on the requirement of the cells. When cells are in need of energy, ADP and acetyl coenzyme can activate citrate synthase enzyme, and if there is too much of ATP is being synthesized but cells don't need that much of ATP because they already have enough, so increase in the cellular ATP level itself can inhibit citrate synthase enzyme. So this is the first reaction of citric acid cycle, which is the synthesis of citrate, and remember that this is the first irreversible reaction of the cycle. Now second reaction of the citric acid cycle is the isomerization of citrate. So in this reaction, citrate is isomerized into isocitrate, and the reaction is catalyzed by the enzyme called aconitase. And remember that citrate and isocitrate, they are isomers, and isomers are basically the compounds with the same number of carbon atoms with different chemical structure. So here you can see that number of carbon atoms, that is 6 carbon atoms, but the location of hydroxyl group is different in citrate and isocitrate. And aconitase, which catalyzes this reaction, is inhibited by a chemical called as fluoroacetate. This fluoroacetate chemical is actually present in rat poison, and that actually inhibits TCA cycle. So fluoroacetate can readily cross both cellular and mitochondrial membrane. So when it is inside the mitochondria, it is converted into fluoroacetyl coenzyme A with the help of enzyme called acetyl-coenzyme synthetase. And this fluoroacetyl coenzyme product is a substrate for citrate synthase enzyme. So what citrate synthase enzyme does is it actually condenses fluoroacetyl coenzyme A with oxaloacetate to form fluorocitrate. So this fluorocitrate is an analog of citrate and is a competitive inhibitor of aconitase enzyme. Therefore, inhibition of aconitase enzyme leads to the accumulation of citrate in the cells. And when there is an accumulation of citrate in the cells, we just learned that increase in citrate level can itself inhibit the enzyme citrate synthase. And hence, it also inhibits the TCA cycle. So basically, rat poison inhibits TCA cycle by inhibiting aconitase enzyme. So this is the second reaction of citric acid cycle, which is the isomerization of citrate. Now, the third reaction of citric acid cycle is oxidative decarboxylation of isocitrate, and this is the second irreversible reaction of citric acid cycle. In this reaction, isocitrate is oxidatively decarboxylated to form alpha-ketoglutarate, and the reaction is carried out by an enzyme called isocitrate dehydrogenase. So the enzyme first basically oxidizes the carbon of isocitrate, followed by the decarboxylation reaction that expels this carboxyl group as carbon dioxide and synthesize alpha-ketoglutarate. So the starting material of the reaction is isocitrate, which has 6 carbon atoms, and the reaction releases carbon dioxide. So the end product, which is alpha-ketoglutarate, has now 5 carbon atoms. So that means the one carbon molecule is lost in the form of carbon dioxide. So if you remember from my previous video that dehydrogenases enzyme promote oxidoreductase reaction, therefore this reaction also concomitantly reduces the coenzyme NAD to NADH. So basically remember that every time there is a involvement of dehydrogenase enzyme, there is going to be a reduction of coenzyme as well. Since this is an irreversible reaction, the isocitrate enzyme is also regulated. So it is inhibited by increased cellular level of ATP, as well as increased cellular level of NADH can also inhibit isocitrate dehydrogenase enzyme. And again, remember that this is the condition which indicates the cells with high energy. On the other hand, increase in cellular ADP level can activate isocitrate dehydrogenase enzyme, and as I have mentioned again and again, that increase in ADP level in the cells indicate the energy deficient status of the cells. And next is also the increase in the calcium content of the cell can also activate isocitrate dehydrogenase enzyme. So overall, ATP and NADH are allosteric inhibitors, whereas ADP acts as an allosteric activator of isocitrate dehydrogenase enzyme. So this is the third reaction of citric acid cycle, which is the oxidative decarboxylation of isocitrate. And now the fourth reaction of citric acid cycle is the oxidative decarboxylation of alpha-ketoglutarate. And this is the third irreversible reaction of the citric acid cycle. So this is the second oxidative decarboxylation reaction of citric acid cycle, which converts alpha-ketoglutarate to succinyl coenzyme A with the help of enzyme alpha-ketoglutarate dehydrogenase. This reaction also expels carboxyl group as carbon dioxide, and therefore you also see that the alpha-ketoglutarate, which has 5 carbon atoms, when it is converted into succinyl coenzyme A, it now has 4 carbon atoms. Now, one important thing to remember about this reaction is the mechanism of this oxidative decarboxylation reaction is very similar to that used for the conversion of pyruvate to acetyl coenzyme A by the help of enzyme PDH complex. However, the only difference between these two reactions is that alpha-ketoglutarate dehydrogenase enzyme is not regulated by any phosphorylation or dephosphorylation reaction, whereas we learned that PDH complex is regulated by phosphorylation and dephosphorylation reaction. And I also mentioned that whenever there is an involvement of dehydrogenase enzyme, there is going to be a reduction of coenzyme, and here in this case there is going to be a reduction of NAD plus to NADH, and the reaction also requires coenzyme in order to synthesize succinyl coenzyme A. Now, since this is also a third irreversible reaction, the enzyme alpha-ketoglutarate dehydrogenase is also regulated by different conditions. First, again, the increase in cellular ATP content and increase in NADH can allosterically inhibit alpha-ketoglutarate dehydrogenase enzyme, as well as the product succinyl coenzyme A, the product itself also can inhibit alpha-ketoglutarate dehydrogenase enzyme. On the other hand, increase in calcium level in the cells can activate alpha-ketoglutarate dehydrogenase enzyme. So this is the fourth reaction of citric acid cycle, which is the oxidative decarboxylation of alpha-ketoglutarate, and most importantly, this is also the third irreversible reaction of citric acid cycle. Now, the fifth reaction of citric acid cycle is the cleavage of succinyl coenzyme A. In this reaction, the high energy thioester bond of succinyl coenzyme A is cleaved to form succinate with the help of enzyme succinyl coenzyme A synthetase. And since this succinyl coenzyme A has this high energy thioester bond, it is utilized to drive the phosphorylation of GDP to GTP. And then the phosphate group from GTP is then transferred onto ADP to synthesize ATP. And most importantly, this is an example of a substrate-level phosphorylation in which a substrate, that is succinyl coenzyme A, provides the energy for phosphorylation to synthesize ATP. Whereas the other way of synthesizing ATP is to transfer the electrons from reduced coenzymes such as NADH and FADH2 to the electron transport chain to synthesize ATP. So there are two different ways of synthesizing ATP. One is through electron transport chain and another is through substrate level phosphorylation. And ATP synthesis by substrate level phosphorylation is the only such reaction in the TCA cycle. So fifth reaction, which is the reversible reaction, is the cleavage of succinyl coenzyme A. Now the sixth reaction, which is the oxidation of succinate. So succinate is oxidized into fumarate with the help of enzyme succinate dehydrogenase. So this oxidation reaction involves the removal of these two hydrogen atoms across the carbon-carbon bond, and by doing that it releases a small amount of energy which is enough to reduce the coenzyme FAD to FADH2. However, the energy which is released by the removal of these hydrogen atoms is not sufficient to reduce NAD to NADH. Therefore, the small amount of energy is sufficient to reduce FAD to FADH2. Now, one important information about this succinate dehydrogenase enzyme. The enzyme is actually the only enzyme of TCA cycle that is embedded in the inner mitochondrial membrane and is also the part of electron transport chain. And we will learn in my next video that how this enzyme play an important role in electron transport chain to synthesize ATP. So this is the sixth reaction of citric acid cycle, which is the oxidation of succinate. Now, seventh reaction of citric acid cycle is the hydration of fumarate, and fumarate is hydrated to malate by the enzyme fumarase, and this reaction involves the addition of a water molecule to the double bond and synthesizes malate. Now, final reaction of citric acid cycle is the oxidation of malate, and malate is oxidized to acetyl-CoA by an enzyme called malate dehydrogenase. And furthermore, the oxidation of malate is coupled to the reduction of NAD to NADH. And malate dehydrogenase enzyme is inhibited by increased level of NADH in the cells. So these are the 8 reactions of citric acid cycle, and now I just want to quickly review the entire citric acid cycle in one slide. So the first reaction of citric acid cycle is the condensation of acetyl-coenzyme with oxaloacetate to synthesize citrate, and the reaction is catalyzed by the enzyme citrate synthase. And remember that oxaloacetate has 4 carbon molecules and acetyl-coenzyme which contain 2 carbon atoms, and when they condenses they form 6 carbon molecules of citrate. And also important thing to remember that this is the first irreversible reaction of citric acid cycle. Now, second reaction is the isomerization of citrate to isocitrate, and the reaction is carried out by aconitase enzyme, which is the reversible reaction. And the third reaction is the oxidative decarboxylation of isocitrate to alpha-ketoglutarate with the help of enzyme isocitrate dehydrogenase, and this is the second irreversible reaction of citric acid cycle. This reaction also reduces NAD to NADH, and it also releases carboxyl group as carbon dioxide, and by doing that the product, which is alpha-ketoglutarate, is now has 5 carbon atoms. Now the fourth reaction of citric acid cycle is the oxidative decarboxylation of alpha-ketoglutarate to succinyl coenzyme A. The reaction is carried out by the enzyme alpha-ketoglutarate dehydrogenase, and this is the third irreversible reaction of the citric acid cycle. And similar to the previous reaction, this reaction also reduces NAD plus to NADH, and it also expels carboxyl group as carbon dioxide. And also remember that the oxidative decarboxylation mechanism of alpha-ketoglutarate is very similar to the mechanism of oxidative decarboxylation of pyruvate to acetyl-CoA. Now the fifth reaction is the cleavage of succinyl coenzyme A to succinate, and the reaction is carried out by the enzyme succinyl coenzyme A synthetase. And this reaction releases coenzyme A when the thioester bond of acetyl coenzyme A is cleaved. And since the thioester bond of succinyl coenzyme A has high energy, it is utilized to phosphorylate GDP into GTP, and the phosphate group from GTP is then transferred onto ADP to synthesize ATP. So this is the fifth reaction. Now sixth reaction of citric acid cycle is the oxidation of succinate to fumarate with the help of enzyme succinate dehydrogenase, and since this oxidation reaction releases small amount of energy, it is sufficient to reduce FAD to FADH2. Now the seventh reaction is the hydration of fumarate to malate with the help of enzyme fumarase, and the last reaction of citric acid cycle is the oxidation of malate to oxaloacetate, and the enzyme responsible for this reaction is malate dehydrogenase, and this reaction also reduces NAD plus to NADH. So in this citric acid cycle, two carbon atoms enter the cycle as coenzyme A and leaves the cycle as carbon dioxide. So this is one important thing to remember. So basically the cycle does not involve net consumption or production of oxaloacetate or any other intermediate of the cycle. The other thing to remember is there are 4 pairs of electrons which are transferred during this entire turn of citric acid cycle. So out of these 4 pairs of electrons, 3 pairs of these electrons were used to reduce NAD plus to NADH, whereas 1 pair of electrons were used to reduce FAD to FADH2. So TCA cycle or citric acid cycle generates 3 NADH. And when these NADH are oxidized through electron transport chain, it approximately synthesizes 9 molecules of ATP. That means 3 ATP is being synthesized from 1 NADH. And the TCA cycle also generates 1 reduced FADH2, and when this FADH2 is oxidized to FAD, it synthesizes 2 ATP. And 1 ATP is also synthesized in the form of substrate-level phosphorylation. So basically the total yield of ATP from the oxidation of one acetyl coenzyme A is 12. That means 12 ATP synthesized per oxidation of 1 molecule of acetyl coenzyme A. So this is basically the overview of citric acid cycle, and I really hope that this is helpful to you and you will learn something new from this. And if you do so, please like and share the video and subscribe the channel. And I'll see you next time with my new lesson on electron transport chain. Thank you so much for watching.